Welcome back, all of you. In this class, I'll start discussing about two-dimensional NMR. An additional dimension for NMR is added to simplify the spectral complexity and to derive information in a better way. So this two dimension has been extended to 3 dimension, 4 dimension, 5 dimensions. So there are n dimension NMR possible. So in this next couple of classes left, I will not be able to complete, cover all 3D, 4D, etc. Even 2D, it's a vast subject in two-dimensional NMR. I will not be able to cover. There are thousands of, several hundreds of two-dimensional experiments possible, which can be designed for specific to your needs. So the goal of this class is not to make you, understand every pulse sequence available in the literature. But in this class, in next 1 or 2 classes about two-dimensional NMR, what I am going to say is what is basic 2D NMR, what are the basic experiments which you require to do when you want to analyze the spectrum of a simple molecule. So I will start with the requirement, how to do a 2D, what is a 2D, how to interpret couple of 2D spectra. I will take examples. We'll go like that. Okay. And of course, in the two-dimensional NMR, the question comes to all your mind, what is a dimensionality in NMR? We're talking 2 dimensions. Then what? You must know something about them. Of course, don't get confused with the physical dimensions. We have always X, Y, and Z dimensions. That's okay. That is different. In the dimensionality in NMR is something different, which we will discuss as we go ahead. First of all, the question one can ask, why we require 2D NMR? What is the need for a 2D NMR? Can't we solve our problems? So since so many classes we have been discussing about varieties of 1D sequences and 1D experiments we can do. We can even enhance the signal intensity, analyze the spectrum. We observed a lot of things. We analyze the in-fact spectrum of many proton spectra and heteronuclear spectra. We discussed in depth of carbon-13 spectra, and we discussed about the editing of the carbon-13 spectra. We understood how to enhance the signal intensity by polarization transfer. A lot of things we understood. Then the question comes is, why we require 2D? What is the use of that? For that, we have to understand some limitations of a one-dimensional NMR. What is the limitation of one-dimensional NMR? First of all, the limitation of the 1D NMR comes because of the size of the molecule that you are interested in. For example, you take a simple molecule ethyl alcohol. What you will do? How many peaks you are going to get? CH3, CH2, OH. Okay, that's possible. Now we know how to analyze those things. That's done. We know what is CH3, where is CH3, CH2OH. How did we make the assignment? Based on the chemical sheet information, using our knowledge of shielding, deshielding, and also we understood a lot about scalar coupling. Looking at the multiplicity pattern, we made the assignment of which is CH3 peak, which is CH2 peak, and which is OH peak. Done. No problem about it. We also analyze this molecule. What is this molecule? A simple molecule. We started analyzing by looking at the multiplicity pattern, triplet, sextet, quintet, quartet, triplet. Again, what is the idea we used? We use the idea of coupling between one spin to another spin. For example, this CH3 was coupled to CH2 and there is no other coupling, long range coupling. So we interpreted this. Remember, in the interpretation of 1H NMR spectra, I took this as an example. So this CH3 will be a triplet because of the CH2 proton. As a consequence, this has to be a quartet because of this. We understood this very clearly. We could interpret, and of course we interpreted sextet and quintet and triplet based on the mixed coupled neighbor, which is the coupled neighbor for each group. For example, for this CH3, the only coupled neighbor is CH2 proton. And if you don't assume there is a long range coupling, the interaction do not extend for a longer range, then we could interpret this because this has to be a triplet because of this. That's correct. We understood all those things. This has to be a triplet and all those things we know what it is. But when we come to this place, we understood one proton is coupled to two protons, one to the left of it, one to the right of it. Okay, we use the logic. This will become triplet of triplet because of this two coupling, and then we understood this will be a quintet, and we know this is a quartet of a triplet because it is coupled to one CH3 to the left, one CH2 to the right. So we understood this. A fairly simple molecule. Now what happens if I give you a very big molecule? Big molecule in the sense, I'll give you a big protein containing, let us say, 40, 50 amino acids. What you will do? Remember, it is not a simple spectrum like this. For example, sometimes each amino acid could be a small molecule of this order, where spectrum can be as complex as this. Now, if I have a protein with 50 amino acids like this, each of them can give rise to C alpha, C beta, C gamma, and NH peak like this. In the entire range of the spectrum from 0 to 10 ppm in the proton spectrum, all 50 amino acid gives proton signals. What you will do? How you will analyze? First analysis is next. First look at the complexity of the spectrum, and it becomes so complex. Resolution is a big problem. You will not be able to analyze beyond a certain molecular size just by 1D NMR. You will simply not be able to identify which peak belongs to which, because severe overlap, severe complexity puts a restriction on the size of the molecule. So as a consequence, you will not be able to analyze the spectrum beyond a small molecular weight size of the order of few hundreds, or sometimes maximum 1000 or like that. Beyond that, if you want to analyze the spectrum of a molecule just by using the 1D NMR, it is very difficult task. So first thing I wanted to tell you was limitation of the size of the molecule. So size of the molecule, as you become bigger and bigger and bigger, there is severe limitation for the use of 1D NMR. I hope you are all with me, right? Now, assignment price is also due to severe complexity. Remember, look at this one. Here I was able to analyze easily. Here is not a big problem. Life is simple. I simply know this is coupled to this, and simply I start making the assignment. Because if you have to get the information about the molecule of your interest, complete molecular structure or conformation, first and the foremost thing is you have to assign each and every peak individually. Every multiplet pattern has to be assigned to a particular proton. And if you know why this multiplet is coming, then you know who is it, who are the coupled partners for it on either side. That is a very important information. So what will happen? As the spectral complexity becomes more and more, as I said, if the molecule size becomes enormously big, you will not be able to analyze so easily. Okay, that is a big point. Assignment is very difficult due to spectral complexity. Second, another important problem comes with the resolution. Here, peaks are well resolved. I know this is a triplet very beautifully. I can identify. You see, if I look at this, if I take the highlighter, I'll show you. This is a very beautiful triplet. This is a triplet, well resolved. This is a quartet. I know it is well resolved. If all of them come, let us say, within this region, all these peaks within this region, triplet, quartet, quintet, sextet, and if you have to identify the peaks when there is severe overlap, how you will do? It is not possible. The resolution is one of the important criteria. There is a way we can enhance the resolution in two-dimensional NMR by using some tricks. Okay, so two-dimensional NMR in fact resolves some information in 2 or 3 dimensions. I have three-dimensional NMR also. In general, it's a multi-dimensional NMR has an ability to separate out the interaction parameters or resolve the interaction parameters or correlate the information in different dimensions, making your assignments very simpler. That is the reason 1D NMR has a limitation. It cannot be done beyond a certain this thing because of enormous spectral complexity. Another thing, let us look at this one. Here, take an example, this sextet. This is coming because of proton 2. Why it is a triplet? Why this is a sextet? Because it is a quartet because of the CH3, and each line of the quartet is split into a triplet because of the CH2. Depending upon the coupling strength, it is a large quartet and each line of the quartet is a triplet. As a consequence, due to severe overlap, you have got this pattern. If this is the case, if the triplet coupling is larger, then it is going to be triplet of quartet. Then the analysis is we have to do it in a different style, and this is a triplet, and each line of this triplet is going to be a quartet like this. Fine. So this is the spectrum if this coupling is larger, CH2 coupling is larger. So it's triplet of quartet, otherwise it's quartet of triplet. That's fine. Now my question is, here when one coupling is larger, I know this is a triplet coupling between this proton to this proton. I know this is a quartet, this separation gives me coupling between this proton to this proton. Because we have been analyzing the proton spectrum since several weeks. We know that. But if there is no resolution like this, they are not resolved like this, it is an overlap like this. Now, how do you know which is a triplet, which is a quartet, and how do you know what is the coupling? It is not possible to, okay, in this case we can still do it, the simple molecule. Let us say this is also coupled to another CH3 or another CH2. Then what you will do? The spectral complexity will be so much because of the multiplicity pattern, they are overlapped. Then the problem, additional problem comes. In addition to identifying which proton is this, you should extract the J coupling of this proton with remaining protons which is coupled to. The extraction of the coupling information is a challenging task. So what you will do for that? Why? Let us think as a possibility. If somehow I can separate out coupling and chemical shift in two different way. I get chemical shift here and remove and get the J coupling here in this direction. And let us I think of two directions. In this direction here I am going to have chemical shift. In this direction, let us say I am going to have only scalar coupling information. If I can separate out, here I will get only J couplings, here I get only chemical shifts. I can make a plot like this. Here I get J couplings and here I get only chemical shifts. What does that mean? I am able to separate out the interactions. Does it not simplify my job? Exactly, that's what happened. This sextet can be simplified if I separate the interactions. You cannot do by 1D NMR. That is one of the biggest limitations. 1D NMR cannot permit you to do this type of separation of interactions. You get the point? That's one thing. So separation of J and delta is a difficult job. And it is not possible to correlate interaction. That is another interesting thing. Here, in this case, what we do is, I know it's a simple molecule, this is coupled to this. I know this is coupled to this based on the multiplicity pattern. In general, in a given by some molecule, there are many peaks overlapped here. Let us say all these four overlapped. Assume all four branch, this thing, multiple pattern, bunch of peaks come here. Everything is overlapped. Now, if you want to identify which is coupled to which, how you will do that? There is no way you can directly do it. But in the earlier days, there used to be what is called a selective decoupling we used to do. What I will do is I will go and do this decoupling by hitting at this proton. When I collapse this, make this energy states of these two these protons saturate, then what happens? This proton will not couple to some of it will break the coupling with some of the, for example, this is proton 4. If I saturate this, it will break the coupling with proton 3 somewhere here. Then this complexity gets reduced. Then I know this is coupled to this. So this is a way we can find out which are the coupled partners, which proton is this sitting next to which proton. The proximity of the proton which is coupled to that one, if you want to identify, okay, nearest neighbor coupled proton, And if there is a enormous spectral complexity, what we have to do is only selective decoupling, which is a very difficult task and also enormous time consuming. It takes, if a big molecule, if there are let us say 500 lines for 300 lines present in the entire range of the spectrum. If I want to get a molecular structure, finding out which is coupled to which, I have to do several hundreds and hundreds of such decoupling experiment, selective decoupling experiment. Then I know this is coupled to this is coupled to this, I can get the structure. This is not possible easily in a one-dimensional spectrum. It's a very, very difficult job. So we have to correlate which interaction, which proton is coupled to which proton. That is called correlation of the interaction to establish the connectivity pattern. For that, we cannot do by 1D, we need to go to 2D. These are all the limitations of 1D. And then forbidden transitions cannot be detected. There is a very interesting thing I told you in NMR. Remember, very first class, one of the classes I said, this selection rule for NMR is that you find out the magnetic quantum number for each energy state and find out the change in the magnetic quantum number between two energy states should be either +1 or -1. Only then I said transitions are allowed. That means where one spin can flip at a time. Of course, there is a combination of spin, three can flip at a time. That is a combination transition, but still the selection rule is one quantum of energy is changed. When spin flips from alpha to beta or beta to alpha, the detected energy is only one quantum. That is called single quantum NMR. Conventional spectra, what you are seeing in NMR spectrum, or conventional is one-dimensional NMR spectrum, single quantum. Okay, conventional one dimension is single quantum NMR. But I showed you in a two spin case example, we have a alpha alpha state, beta beta state, alpha beta beta alpha. All four energy states are there. I said this magnetic quantum number, total magnetic quantum of this energy state, beta beta state is -1, alpha alpha state is +1. Can there be a transition between these two states, alpha alpha to beta beta? It is forbidden transition because it requires double quantum, two quantum of the energy. Single quantum requires only one quantum of energy, one quantum, because only one spin can flip alpha to beta or beta to alpha, but here requires two quantum of energy. Or if I have a three coupled spin system, I can I get the transition from this and lowest energy level alpha 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 to beta beta beta. If I want to see that transition between two energy states in the three coupled spin system, three quantum separation, it is not possible. They are all forbidden transition. These are called multiple quantum transitions. These are all forbidden. You cannot detect in a conventional one-dimensional NMR. But of course, in two-dimensional, indirectly there is a way to detect. We can do that. It is not difficult at all. So this is one biggest advantage, limitation of the. And what is the use of detecting such forbidden transition, multiple quantum transition? Enormous advantages there, which I am going to tell you. Many information can be derived. First, we can simplify the spectrum. We can analyze the spectrum in an easy way. Lot of advantages are there. We can do spin system filtering. Lot of things we can do by detecting multiple quantum spectrum. That is one of the limitations here for an 1D NMR. So 1D NMR has several limitations. Another limitation is simultaneous detection of two different nuclei is not possible. See, in one dimension NMR, I can see proton NMR. I can detect proton signal. Send a RF pulse, 90 pulse on proton, bring the magnetization to XY plane, start collecting the field-intention decay and get the NMR spectrum. Fine, the proton spectrum. If I want to do the carbon-13, okay, we can do that. I'll apply carbon 90% the carbon channel, bring the magnetization to XY plane, start decoupling it simultaneously immediately after the pulse and start collecting signal. You get decoupled carbon-13 spectrum. But in one experiment, if I want to get carbon chemical shift and also proton chemical shift simultaneously, it's not possible. I cannot detect it. But On the other hand, if I go to two-dimensional NMR, there's a big advantage. I can do a heteronuclear two-dimensional experiment where I can get carbon chemical shoot in this direction, proton chemicals in this direction. I can get heteronuclear coupling in this direction and chemical shoot of proton or carbon in this direction. So all such interesting things we can do in 2D NMR, and in 1D NMR all these things access are limitations. That is the reason why 1D NMR cannot be employed for many things, especially as the size of the molecule keeps increasing, and for various things, problems which I suggested here, 1D NMR spectrum has lot of limitations. Are you all with me? I hope you are all with me, right? Okay. This we discussed. Now, as a consequence, very interesting discovery took place. Thanks to all our seniors, pioneers in NMR spectroscopy, who made a breakthroughs in this research. As a consequence, we can utilize NMR the way we want today. This is because of many stalwarts pioneers who made a significant contribution by doing a lot of discoveries. And this two-dimensional NMR discovery, of idea of this was given in 1971 in a Ampere summer school, Yugoslavia, by Jenner. Jenner presented a paper in summer school in 1971 where he gave the idea of two-dimensional NMR. What is two-dimensional NMR? And that we'll come to that later. That was called rebirth of NMR. Till 1970, from the year it was discovered, first observed in 1945, NMR was used to some extent after the discovery of chemical shift in 1945, 49, and then J coupling. Slowly everything was observed, and then NMR was used to some extent. But application of NMR to a very big molecule like a biomolecule was greatly restricted. Largely it was restricted till 1970 because, again, as I said, you have to use only 1D NMR, which has several restrictions. A lot of constraints were there. But 1971, because of the advent of two-dimensional NMR spectroscopy, which was reintroduced, we call it as a rebirth of NMR. NMR spectroscopy took another birth. Thanks to Professor Richard Ernst, a Nobel laureate who contributed for this, who is the one who is responsible in early 80s and early 76, also first paper in 1976 published in Journal of Chemical Physics here. And this is a key paper of 2D NMR. Afterwards, literally it took off in the early 80s. He published the paper in 1976, first paper on the idea of two-dimensional NMR. Literally after 80s, it has, just exploded like anything. And lots and lots of pulse sequences were designed. Lots of pulse two-dimensional NMR experiments were designed and modified to derive specific information. And literally the application of NMR by utilization of two-dimensional NMR, then it extended further to multi-dimension, like 3-dimension, 4-dimension, everything began. And that's where the application, utility of NMR and the idea of NMR, development of NMR, everything exploded during that time. And that's why NMR has become very powerful now, because we can use it in a number of possible ways. Okay. So with this, now I will ask a question. You may ask another thing. Fine. What do you mean by this two-dimensional NMR? Why really we require it? Whether we require two-dimension or higher dimension? How do I know? You may say there is a limitation of 1D NMR, but there are many higher dimension experiments, 2 dimensions, 3 dimensions. You may ask me a question, which one I have to use? What experiment I should think for my molecule? That depends upon, look at the spectrum like this. It is ubiquitin with 76 amino acid, 8.5 kilodalton molecular, right? And look at this range. This is a very old spectrum. Nowadays, in a high frequency, you may get a better spectrum. Look at this region. You can only assign side chains here and methyl here, aliphatic here, C alpha here. Where is the resolution? It's a big problem. Here, more than thousands of peaks here. All resonances have been squeezed into 10 ppm range, that is the range of chemical shift range of protons. The proton chemical dispersion is in the range of 0 to 10 ppm. So all 76 amino acid, each of them can be considered a spin system. 76 spin system with so many protons present. All the peaks are hidden here. Just look at the spectrum of your molecule depending upon the molecule size. Then you have to decide which one to use. As a consequence, we require to go to higher and higher dimensions. Now, what is that higher dimension? What do you mean by dimensionality? And which molecule requires which dimension is the next question. Let us answer. I hope you are all with me, right? Now, what is this NMR spectra pulse sequence what I have written? All of you must be knowing by now. This is a conventional one-dimensional spectrum. Apply a radio frequency pulse. Maybe I'm looking at proton. Apply a one-dimensional pulse on proton channel. Start collecting the signal. Of course, this is called a dead time delay given. Don't worry. It can be given. It need not be given. In principle, without delay, you have to start so that there won't be any phase problems. In the NMR spectrum. So you have to immediately start collecting the signal from the 0 point. Okay, doesn't matter. This is the one-dimensional pulse sequence where you apply a pi beta pulse, 90-degree pulse, bring the magnetization to XY plane and start collecting the signal with your detector or receiver is here. Fine. This what is happening here now. If you look at it, you are collecting the signal as a function of time here. Okay, you are collecting the signal as a function of time. And whatever you are going to do is you will do the Fourier transformation here. Remember, I said when you collect the signal in time domain and do the Fourier transformation, time and frequency are Fourier pairs, I said. Mathematically, time and frequency are Fourier pairs. Now what is happening is you are converting the time domain signal to Fourier frequency domain signal. What you are going to get is spectrum like this. So this is our one-dimensional NMR spectrum, where we assigned all the peaks and then got the area of the peaks, multiplicity, etc. Now here we can divide this into some time zones, okay, some time periods. One-dimensional spectra in reality is two dimensions. You know why? In this dimension we get frequency. In this dimension we get the intensity, but we don't bother so much about it. We always talk dimensionality in terms of time. In time here, we are collecting the signal as a function of time. How many time domains are there here? Only one time domain. In a single time domain, you are collecting the free intention decay and doing the Fourier transformation. This is called one-dimensional NMR because single time period is there. Single Fourier transformation gives you frequency spectrum like this. Okay, I will call this is the relaxation delay. This is called a preparation period. Okay, and this is where spins are allowed to relax and attain thermal equilibrium. And afterwards, you apply a RF pulse, collect the signal here, and I call this as a detection period. Okay, so 1D NMR, what is happening? We have only one time period. It's a of course, two time periods, I would say. One is a preparation period where you don't do anything. You simply keep quiet. Put the sample in a magnetic field and keep quiet. Allow the spins to attain thermal equilibrium. There is a build-up of bulk magnetization. Simply keep quiet for some time till it builds up. That's all. That is called preparation period. Time period, also called relaxation delay, for the spins to attain equilibrium. Then apply pulse and collect signal. This is a real time domain signal. We are collecting the signal and do the Fourier. This is where we are getting the real signal from the sample. Okay, signal is collected in one time period here and do single Fourier transformation and get a frequency spectrum. That is why it is called one-dimensional NMR, 1D NMR. You understand? What is the dimensionality? Dimensionality refers to time. Time domain. How many time domain periods are there for you? You are collecting signal. You are collecting signal in one time domain. It is a single quantum. If I collect two time domains, it is two dimensions. If I collect signal in three dimensions, we'll see that later. And general pulse sequence for a two-dimensional NMR consists of 4 time periods. Okay, what are the 4 time periods? First is a preparation period like this. You understand? First, and oh sorry, and we have a one pulse here. We apply a pulse here. Okay, pulse also we can talk later. There are 4 time periods we talk: preparation period, evolution period, mixing period, and this is called a detection period. It's a real period at which you collect the signal for doing the Fourier transformation. In principle, we have 2 time domains here. This is one time domain where we allow the fields to evolve. This is another time domain where we collect. This is the constant fixed time, constant time. This time for which you collect signal is not varied, but this time domain, this time is varied. Now we collect the signal here and do the Fourier transformation. Here indirectly, here also we collect the signal. Similar to FID, I will tell you how we do it later. Here you do something for the spins in the preparation period. Allow the spins to evolve in this period. How it evolves? Evolution takes place like human evolution, like evolution in the life. Whatever you do for the spin system here, with do something for the pulse sequence, bring it here, and it starts evolving with time as a depending upon type of interaction. It could be J coupling, chemical shift, homonuclear, heteronuclear J coupling, whatever it is. Some parameter is responsible between two spins, which two coupled spins or interacting spins. As a consequence, the spins will start evolve with time. They behave. Their behavior pattern changes with time here. Okay, crudely. And then what happens is whatever happens here is reflected here. We are going to collect this signal in this time domain and do the Fourier transformation. We collect signal here and collect signal here, do 2 time signal. If I have 1 time domain signal, I will do one Fourier transform. If a 2 time domain signal, we have 2 Fourier transformation. Double Fourier transformation I can do and get the frequency spectrum in 2 dimensions. Like 2 dimension time domain signal gives you 2 dimensional frequency domain signal. That is the double Fourier transformation. So we will come to that how we do that later. And for the day, I will stop here. I wanted to introduce generally the pulse sequence for a 2D NMR. They have not completed yet. We will start from this again tomorrow and continue further.